Hello and welcome everyone to this podcast for genetics. This is going to be the first of several podcasts where we talk about mutations. But before we start talking about mutations, I want to spend a little bit of time talking about where we've been and how that's leading up to our discussion for mutations. Remember, at the beginning of the semester, we began by talking about nucleotides. Guanines, cytosines, uracils, thymines, and adenines. And then that led us to our discussion on DNA and how that was the genetic material and all the evidence that supported that DNA is usually the genetic material. And then we talked about how DNA can copy itself through replication. We then moved to how the DNA here, we also talked about a lot of other features with DNA, how it was structured, how it could be packaged, and all that good stuff. We then quickly moved into talking about how specific regions in this DNA contain genes, and how these genes could be transcribed to make RNA molecules. And then how these RNA molecules could then be translated into proteins. And all the steps that go in this process of going from RNA to protein. And we talked about how this protein often is the trait, the characteristic, the thing that we see. I want to come back over here. We also talked about how that DNA is replicated, how that's packaged into new cells. And you reviewed mitosis, and we talked further about meiosis in this process of making new cells. For the most part, what we talked about here is what usually happens. Every now and then we talked about a mutation to explain something, but in general, we're talking about how we go from this DNA molecule to a protein. But now what we want to talk about is how, when we change the sequence on the DNA molecule here, how that can lead to new proteins or new functioning proteins. Sometimes new proteins that have a beneficial effect, or sometimes it may generate a protein with a negative effect. What we're going to spend the next several podcasts doing is talking about how this DNA, sometimes through the process of replication, sometimes through the process of mitosis and meiosis, will generate mutations. Okay, now I want to talk about the two broad categories of mutations we're going to spend the next few podcasts talking about. The first is at the chromosomal level. And the second is at the DNA level. But what I mean by at the DNA level is that this sequence in the DNA, this series of G, C's, T's, and A's in the DNA, codes for a very specific protein. And at the DNA level, when we change the sequence, so why don't we just write that here? A change in DNA sequence. That's what we're talking about here. How that change in sequence is going to lead to a different protein. Mutations at the chromosomal level, which we're going to talk about first, are much broader in scope. Now, regardless if we're talking about a chromosomal level mutation or DNA level mutation, we can think of mutations as having one of a few effects. First, sometimes you can have a mutation and it has no effect. Sometimes it can create a non-functional protein. And sometimes this mutation, we can think of it as creating a new function. Probably not a brand new function that's never been seen before, but some sort of improved function compared to what was seen in the past. And these mutations, particularly the new function ones, are really a major source of evolution. All right, so before we end this podcast, I want to spend some time introducing chromosomal level mutations, because we're going to spend the next several podcasts focusing in on these. All right, so chromosomal mutations. To introduce chromosomal mutations, let's look at this karyotype that has come from a healthy individual. We can lump chromosomal mutations into two broad categories. The first include a change in chromosome structure. These include deletions and duplications. A deletion would not be a deletion of the whole chromosome. Rather, it would be a deletion of a part of the chromosome. So maybe this would be removing just that one end there. Likewise, a duplication would not be duplicating the whole chromosome, but rather maybe you add an extension to one of the chromosomes, make it larger. The other two are translocations and inversions. Translocations are when you, say, take this part of chromosome 1 and you move it to this part of chromosome 2. Inversions, on the other hand, are when you take, who should we pick on? We'll pick on chromosome 4 here. Inversions occur when you cut out a portion of the chromosome and flip it around 180 degrees, so that initially one end of the chromosome was on this side, but now it's on this side. The other type of chromosomal mutations result in a change in chromosome number. These could be aneuploids, and we'll talk more about aneuploids later on, but aneuploids would do one of two things. They may add a whole extra chromosome. Not all the chromosomes, but in this case, just chromosome 15. Or you probably have heard of Down syndrome. Down syndrome is when you have one extra copy of chromosome 21. Or sometimes an aneuploid removes a chromosome. So maybe you remove all of chromosome 20, one of them. You still have the other one. So in humans, for instance, we usually have 46 chromosomes. But aneuploids can cause that number to go to 47, as in the case here where we added a chromosome, or it can make it drop to 45, in this case when we lost chromosome 20. The other type of a change in chromosome number we're going to call a polyploid. A polyploid is when we add one extra chromosome to each set of chromosomes, just like I'm showing here. You could also add more than one, but I just showed the one. So you could end up then, instead of having 46 chromosomes, you would have 69 chromosomes. In the next podcasts, we're going to talk about deletions and duplications, and then we'll talk about translocations and inversions, and then we'll close with aneuploids and polyploids. And there's one other thing we'll talk about towards the end here, and that is how aneuploids and polyploids form. Okay, that's all I'm going to say on this podcast. But if you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.